So, something I think interesting in virology is the differences between plant and animal viruses, because all viruses tend to be somewhat similar, but these ones are interesting, especially in how they infect and leave cells. So, animal viruses, they are usually enveloped, and I'll explain what that means, but they enter the cell, they leave the envelope behind, they do their replication and everything, and then as they're leaving, they have to bud through the host cell membrane, and doing that, they obtain their envelope, which is just whatever the host cell membrane is made of. And in this case, almost always their envelope is made of the phospholipids that make up the animal cell outer membrane. So in order to obtain their membrane, they literally just steal it from the host cell. They're just like, this is mine now, thank you very much. Whereas in plant viruses, plants have this thick cell wall that is very difficult for viruses to get through. And so almost entirely, plant cells are not enveloped, and also they have to enter through imperfections in those plant cell walls. So for example, if the plant is damaged, the virus can get in through the break, or they very, very often use vectors for transmission, such as insects. When an insect, for example, an aphid, is on a plant, it is eating that plant, and the virus is transmitted from that aphid, where it's just hanging out, into the plant through whatever holes the aphid made in those plant cell walls. And then when leaving the cell, it does similar things. It will exit through damage, or it will also use motor proteins to move it to the plasmodesmatas, which are junctions in between plant cells, and infect new cells through those. But they can't enter a cell initially through plasmodesmata. There has to be pre-existing damage or damage done by a vector in order. I got distracted by my roommate and got a granola bar and completely lost my train of thought. But anyway, so animal viruses, they yoink the envelope from the cell membrane of their host. Plant viruses, they need to be put into the plant cells by damage or something because they can't get past the cell wall. So, yeah, there we go. I lost everything I was gonna say.